Welcome to Suhail's Paramedical and Nursing Academy. Welcome to Suhail's Paramedical and Nursing Academy. In the previous lecture, I was explaining about the mitosis. In mitosis, actually, we was explaining about the cell cycle and cell division. In cell cycle and cell division, I have explained about the mitosis. Actually, cell cycle and cell division means it is a process. It is the process of cell division. Cell division means the cells divide. The simple means of cell division means cells will divide in the body. Why cells are dividing in the body? The reason is clear. Suppose in case of when the person is when in case of fertilization, there is a need of ovum as well as sperm. So the need of ovum and sperm means they should first form. The formation of ovum and sperm will occur by means of both mitosis as well as meiosis. Similarly, whenever there is what happens, whenever there is any kind of injury, whenever there is any kind of trauma, whenever body needs growth and development, that time also mitosis plays an important role. So mitosis is the main process that is shown as the main division. Mitosis is the division that is shown by the primordial germ cells as well as mitosis is shown by the somatic cells of the body. This is all about the mitosis. In the previous lecture, I have explained about the cell cycle, the setups of cell cycle, in which I told there is interphase. Then I told there is after interphase. What I told in interphase in cell cycle there is interphase, there is M phase. M phase I have explained that is mitotic phase. In interphase I told there are G1, S, G2 phase. In case of mitosis, I told there is prophase, metaphase, anaphase, telophase. What occurs, what happens in these steps, I have explained very clearly. And suppose if any person has not understood well, then first see that lecture, then only he can come to this. Directly coming to meiosis, it will not be beneficial for that person, because in this today I am going to explain about the meiosis. Because in case of meiosis, I will leave those steps which I have already explained. Okay, I will take only that part of meiosis which is important, which is different from the mitosis. Okay, in case of mitosis, what happens? Actually, cell division occurs by two ways. Either every in cell cycle, every cell takes first. Whenever a cell is going to divide, whether the cell is a germ cell, whether the cell is a somatic cell, whatever the cell is, first it will show interphase. So cell cycle, the common phase is interphase, that is G1, S and G2 phase. So G1, S and G2 phase already here. So G1, S and G2 phase already here. Okay. Then after the cell completes G1, S and G2 phase, now it will enter towards the M phase. Now there is a question. If it is a primordial germ cell, or if it is a somatic cell, it will enter into the mitosis. That I have already explained. Now another question comes, if it is not a primordial germ cell, after primordial germ cell divide, it forms oogonium. Suppose in case of humans, it will form oogonium and spermatogonium. This oogonium and spermatogonium shows after interphase, it will show meiosis. So in case of meiosis is seen in case of germ cells. Why are those germ cells present? Actually, in case of males there are testes, in case of females there are ovaries. So the ovaries and testes have role in oogenesis and spermatogenesis respectively. Okay, so germ cells, these are called as the germ cells are present in the testes and ovaries. Okay, the germ cells means, suppose there will be oogonium. In case of female, there will be oogonium, or if it is male, the oogenesis, the process is called as oogenesis. So oogenesis is shown by the oogonium. Okay, spermatogenesis there will be formation of sperm. Okay, it will be shown by what? Spermatogonium. So the oogonium and spermatogonium enters into a specific kind of division, a special kind of division. That cell cycle division is called as meiosis. Okay, so it is the meiosis that is shown by the germ cells. Remember, so this meiosis is responsible for the formation of sperm, for the formation of ovum. Okay, so the variations in the genetics. Now we will enter towards the genetics. The variations that comes in the variations that comes in the generation to generation is because of the meiosis. So meiosis is important step in the cell division. It is the important step that occurs in case of germ cells. That occurs in case of germ cells. So meiosis is, that is why if you see that the evolutionary variations comes in the meiosis. Okay, so meiosis, the variation is a normal process. Remember, variation means variation is a normal, but mutation is not a normal thing. Mutation means when there is any deviation is coming from the normal genome, that is mutation. But here there will be variation. The variation is beneficial for the human race, for the human species. Whenever there is a variation, then only there will be the variety of characters, variety of behaviors in the human population. That I will tell later where that occurs. So today we are going to explain about the meiosis that occurs in case of oogonium, that occurs in case of spermatogonium. So what will happen? What will happen in meiosis? First I told in cell cycle starts from interphase. Interphase will be same. So now cell will, after the interphase, cell has increased its DNA content. I already told G1 phase, growth phase. S phase, DNA content, central duplication will be there. And in case of G2 phase, the cell will show further growth, whatever requirement is there for the future. Okay. Now I enter towards the meiosis. Now I enter towards the meiosis. So in case of meiosis, what will happen? In meiosis, there will be two processes. One is called as meiosis after interphase. After the interphase, the cell will enter into meiosis first. Meiosis first. Then after meiosis first, it will enter into meiosis second. The meiosis second is same like mitosis. Okay, so mitosis and meiosis second is same. Remember the point. But the mitosis and meiosis first is not same. So meiosis, there will be first meiosis first, then meiosis second. This meiosis first, what is this? It is also called as meiosis first is also called as reductional division. It is also called as reductional division. Why it is called as reductional division? Remember, it is called as reductional division because there will be the half decrement of chromosomes in the daughter cells. So from one cell there will be formation from the oogonium, there will be formation of two primary spermato. Okay, so oogonium will enter into meiosis first. Meiosis first will occur in the primary oocyte. The primary oocyte will enter into meiosis first. So the meiosis first is completed by the primary oocyte. So what will happen when these spermatocytes, when these germ cells will enter into meiosis? What will happen after the interphase? They will enter into meiosis first. The meiosis first is called as reductional division because the content of DNA will be reduced to half. Okay, how it will reduce? I will tell. Second, it is also called as heterotypic division. Why it is called as heterotypic division? I told in case of mitosis what happens. I told in case of mitosis there is 2n. What happens in case of mitosis? I told that from 2n there will be formation of 2n. That means the chromosome number will remain same. The chromosome number will remain same. Okay, as well as here will be also father's copy, mother's copy. Here will be also father's copy, mother's copy. So 2n means from 2n it will be 2n. 2n means both father copy. When cell will divide, it will form two cells. This cell will have both father's copy as well as mother's copy. This cell will also have one father's copy of chromosome, one mother's copy of chromosome. That's why it is 2n. So both mother's and father's copy will be there. But in case of meiosis, what will happen? There will be formation of two daughter cells. But in two daughter cells, one will be having the father's copy only, another will be having the mother's copy only. So it is n. You can understand by this way. So mother's copy only is there, it is n. Father's copy is only there, it is called m. So when we join these n and n, then it will form 2n. But there will be half chromosome. It will be 23 chromosomes will be there. Here will be also 23 chromosomes. That is reductional division. That I told reductional division. From 26 chromosomes it will form 23. Both will be having 23 chromosomes, and this cell will be having only father's copy, and another cell will be also having mother's copy. That is called a heterotypic division. That is why it is called as heterotypic division. So these are the two important points that I told here. Now I will explain the meiosis. So in meiosis, interphase already explained. There will be meiotic phases. In meiotic phase, there will be meiosis first and there will be meiosis second. The meiosis second is same like mitosis. I will not explain here. Mitosis second, meiosis first you have to understand. Then mitosis, I will tell you what will happen in the mitosis second in a quick revision. In meiosis, in the meiosis first, there will be many stages. In case of mitosis, there was also many stages. Here will be also same stages: prophase, metaphase, anaphase, telophase. Here will be prophase first, metaphase first, anaphase first, telophase first. Okay, but the prophase first here is the complex phase. It will be consisting of 5 phases: leptotene, zygotene, pastine, diplotene, diakinase. If you understood these 1, 2, 3, 4, 5 steps in case of prophase first, it means you have understood the meiosis. So the thing is, you have to understand clearly prophase first. After prophase first, there will be metaphase first, anaphase first, telophase first, metaphase first, anaphase first. Okay, prophase, metaphase, anaphase, telophase. Okay, so metaphase first, anaphase first, telophase first. After the prophase first. In prophase first, I am saying that there will be first say leptotene. What will happen? I already told in case of interphase, the DNA in the interphase, the DNA will be less condensed stage. It will be in the hydrated form. It will be in the hydrated form, less condensed, and it will not be visible in the cell. That will be in case of interphase. That is why it is called as chromatin. In case of interphase. In case of M-phase, I told it will be condensed form of DNA, dehydrated form of DNA, a thicker form of DNA, clearly visible, and that is called as chromosome. Okay, so now we will talk about the meiosis first. In meiosis first, I am explaining about the what prophase. So in prophase, in prophase there will be the formation of what? There will be in prophase there will be many steps. Prophase first, many steps. These steps are not found in the mitosis. Remember, in prophase first, the first stage is leptotene. In what will happen in the leptotene? See here, in leptotene, this is the father's copy. This is the mother's copy. The father's and mother's copy is not so much condensed. It is not so much condensed. This is less condensed in this stage. Okay, and this leptotene stage is called as thin thread stage. Thin thread stage. It is called as thin thread stage. Why it is called as thin thread stage? Because the chromosomes will be like a thin thread, like a threads, very thin and less visible. And what will happen? These chromosomes, father's copy, mother's copy, these chromosomes will be arranged like this in the cell, and they are not very clearly visible. Remember the point. In the microscope, we are seeing that this chromosome will be touching on the nuclear membrane. This chromosome is also touching on the nuclear membrane. These threads are touching nuclear membrane. There will be one centromere. It is also having centromere. It is also having centromere. Okay. These are called as homologous chromosomes. This is homologous chromosome. This is homologous chromosome. And remember one thing, that the telomere means the end of the chromosome is attached to the nuclear membrane of the cell. It is showing attachment towards the nuclear membrane of the cell. That is why this process is called as synthesis. This is called as synthesis. So I have written here, this is called as thin thread stage because there are thin thread-like structures are there. And remember, there will be like this is centromere, main centromere, and there will be some beads, some bead-like appearances. That is why it is called as beaded appearance. So there will be the beaded appearance. The bead-like form will be like a bead. So suppose what is bead? This is a stick. Okay. One thing here, one dot. This is a bead. This is called a bead. So beaded-like appearance will be there, and the condensation. Remember, this beaded-like appearance will be due to many structures. These are called as chromomere. I have written here chromomere. It will be due to the chromomeres. These the attachment, these the structures which form a beaded appearance is called as chromomere. It is called as chromomere. Many chromomeres will be there on the chromosome. This is called a thin thread stage. Beaded appearance will be there, and the condensation of chromosome will occur in this stage. And it is also called as vacuole stage. Why it is called as vacuole stage? Vacuole means there will be arrangement like a vacuole. There will be arrangement like a vacuole. See, this is touching the nuclear membrane, this is also touching the nuclear membrane. When all the chromosomes will touch the nuclear membrane, they will form like a vacuole. Like a vacuole. B-A-Q-U-E-T. Vacuole arrangement. Okay. Another stage is called as zygote. So condensation will be increasing. The condensation will increase from lactotine, zygote, pagotine, diprotein, diakinesis. It is clear. Condensation will occur more and more. In zygote, so more condensation will occur. This zygote is called as pure thread stage. It is called as pure thread stage. Why it is called as pure thread stage? See here. This is called as, zygote is called as pure thread stage. These chromosomes will homologous chromosomes. This is the father's copy. This is the mother's copy. These are called as homologous chromosomes, and the sister chromatids of the sister chromatid of this chromosome is not visible. Remember, the sister chromatid of this chromosome is not visible. That is why it will be in the bivalent form. There will be the form in bivalent. Bivalent means only two centromeres are there, one and two centromeres and two chromosomes. So there will be no sister, only two chromosomes, two centromeres. Okay. 2 centromere, 1 and 2 chromosomes, but sister chromatid is not visible. Only two sister chromatids are there. That is why it is called bivalent. Bivalent means only two non-sister chromatids are visible. Only two non-sister chromatids are visible. That is why it is called a bivalent stage. This bivalent stage is zygote, and pure thread stage means they will be in the pure form. These homologous chromosomes will come close to each other. They will come closely like a pair. So that is called a pure thread stage. So pairing of homologous chromosomes will occur. The pairing of homologous chromosome, that means the father's copy and the mother's copy. The pairing of homologous chromosome is called as synapse. This pairing of homologous chromosomes, it is called as synapse. It will occur. This synapse occurs because of a special protein. That protein is called as synaptonemal complex. Okay. So synaptonemal complex is a complex of proteins. That helps in the pairing of two homologous chromosomes towards each other. Okay. So you have to remember synapse will occur in the zygote stage. Okay. Then there will be the pagotine stage. So in this lactotine, chromosomes was less condensed. They was touching the nuclear membrane in lactotine stage. Now in zygote stage, condensation occur more and more. Now there will be visibility of two homologous chromosomes, but only two non-sister chromatids will be visible. That means one sister chromatid will not be visible. That is why it is called as bivalent stage in the zygote. Okay, so these only two will be visible. Then what happens? They will come close to each other. The process is called as synapse. The pairing of homologous chromosomes towards each other. This is called as synapse. Then what will happen? After two chromosomes come close to each other, what will happen? You have to see towards the pagotine stage. They will enter into the third stage. In pagotine stage, third stage, more condensation of DNA will occur. But in case of pagotine stage, what will happen? Remember that point. Already I told chromosomes come close to each other. That is called synapse, which occurs in the zygote stage. But in pagotine stage, when they are already close to each other, there will occur a special process. The special process is called as crossing over. Crossing over means it has been explained by this crossing over here. It is explained here. This in pagotine stage, that is called as breakage and reunion hypothesis. The breakage and reunion hypothesis says that there will be the breakage of DNA between the two homologous chromosomes. That means, if we say in clear words, that means between the two non-sister chromatids. This is one chromatid of this chromosome, chromatid of this homologous chromosome. These are each other. I told already in the previous class. They are called as The two sister chromatids of the same homologous chromosome. The chromatids of same homologous chromosome is called as sister chromatids. But here, non-sister chromatids will show crossing over. What is crossing over? There will be the breakage of DNA in one non-sister chromatid, and that will link to this. That is called as breakage. That is reunion, and it will attach to the another side of non-sister chromatid. Then that part will enter into this. So there will be breakage of the DNA, some part of DNA. Then reunion will be there. They will connect in the see what happens. Here they have see here I have shown the clearly how the crossing over occurs. So crossing over occurs in the Pagetti stage. I have already told here in Pagetti crossing over occurs. And one more thing, there was only bivalent. Here will be tetrad. Tetrad means there will be the visibility of two homologous chromosomes, and there is presence of two centromeres. But there will be the appearance of four chromatids, two sister chromatids in one homologous chromosome, two sister chromatids in another homologous chromosome, which are found in the Pagetti stage. Okay, so crossing over, tetrad, further condensation I told will occur in this stage. And remember the point, the important point is this crossing over, this breakage and reunion of DNA between the two non-sister chromatids occurs by the help of an enzyme. The enzyme is called as recombinase complex. Enzyme recombination complex. So recombinase enzyme complex is a special kind of enzyme which helps in the recombination. That is, recombination means what? The crossing over is also called as recombination. The crossing over is also called as recombination. That is why the enzymes which help us in the recombination, they are called as recombinase enzyme complex. The recombinase enzyme complex helps us in the recombination, that is crossing over of the two DNA between the two non-sister chromatids. So remember the point, when there occurs the crossing over between the non-sister chromatids of two homologous chromosomes, when this crossing over occurs, remember it will create variation in the generations. The variations that you find in the species, in the generation, those variations are due to the crossing over. They are due to the crossing over. Another thing is, after the Pagetti stage, what will happen? There will be another stage that is called diplotin. In diplotin stage, actually already the crossing over is going on. Crossing over between the two non-sister chromatids are already here, but this crossing over will not be visible in this stage. It will be visible in the another stage that is called as diplotin stage. A X-like structure will be visible. A X-like crossing will occur. This is X-like structure is visible. This X-like structure is called as chiasmata. This X-like structure is called as chiasmata. This chiasmata will be visible in case of diplotin stage. Okay. So diplotin, it is called a double thread stage. In diplotin, it is called a double because double threads are there. The two homologous chromosomes forming X-like structure, that is called as chiasmata. It occurs in the diplotin stage, and in this stage, now the homologous chromosomes, the non-sister chromatids of homologous chromosomes which have shown already the crossing over, which have already shown recombination, they will try to detach each other. They will try to detach, that occurs in the diplotin stage. So there will be termination. So termination will try, there will be the startage of termination. So two homologous chromosomes will try to go away from each other, but they are not going now. They are trying to go away from each other. That is called as termination of diplotin. Another stage will be there, that is called diakinesis. In diakinesis, what will happen? In diakinesis, what will happen? These non-homologous chromosomes who have already shared the DNA, who have already shown breakage and reunion, that is crossing over, these homologous chromosomes will go away from each other. There will be the division between the two homologous chromosomes, that is called as termination. So complete termination will occur. The complete termination will occur in the diakinesis stage, and the DNA will be more condensed in this stage. This is called as diplotin. What will happen? So already variation occur, already crossing over occur between the two sister chromatids, between the two non-sister chromatids. Okay. So in metaphase, first what will happen? Say I told in the mitosis, see here, I told this was the mother's copy. sorry, see, this is one copy, this is one copy. Okay, this is one chromosome. In case of mitosis, there was arrangement of another chromosome like this way. So here also spindle fibers are attached. Here also, this side also, this side. So because this is a cell, this side also spindle fibers will attach. This side also will attach. This was in case of mitosis. But in case of meiosis, first what will happen? This homologous chromosome will be here. Okay. So the spindle fibers will attach only one side of the homologous chromosome. Only one side of the homologous chromosome. So there will be no detachment of centromere. Centromere will not divide. There will be no division of centromere. So one father's copy will go one side, mother's copy will go another side. So what happens? It will create N. It will create N. I hope you understood. Because now once more I will say here. What will, this was metaphase. They annihilate in the center. How? Like this way. Metaphase. This is the father's copy. This is the mother's copy. Spindle fibers will attach on the both sides on only one side of the homologous chromosome. So this chromosome will go this side. This will go this side. Only one side. So father's copy will go one side. Mother's copy will go another side. Okay. So when it will go, I will tell. Firstly, there is attachment of spindle fibers. In case of anaphase, first this separation will occur. This separation will occur. So in case of mitosis, there was division of centromere. There was division of centromere, but here division of centromere will not occur. Okay, this will go one side in anaphase first. This will go anaphase because in anaphase division occurs. Then in telophase, decondensation will occur. This DNA will undergo decondensation. See, anaphase I have shown. This is already gone. That side. This is already gone. During the depolymerization of spindle fibers, this is going this side. Father's copy one side, mother's copy. So it had created the two cells. The two cells will be formed in the after the in the telophase. There will be decondensation of DNA. Now there will be the formation of nuclear membrane in the telophase. First, what I told already in the mitosis. Same will occur in the telophase, what occurs in the mitosis. Okay, but here what will happen? Two cells are formed. In two cells, there is father's copy in one cell, mother's copy in one cell, and which are having the variable DNA that occurs during the pachytary stage of prophase first. Prophase first pachytary stage. The crossing over occurs. That crossing over has created variation. That is here is attachment of DNA from one non-sister chromatid into the another non-sister chromatid. This was all about the meiosis first. What will happen? So two cells are formed, one and one. That is why it is called as heterotypic division. Heterotypic. Why it is called as heterotypic? Because two cells are there. This cell will be also having 23 chromosomes. This cell will be also having 23 chromosomes. But this cell will be having father copy. This cell will be having father copy. Father copy. This will be having mother's copy. That is why it is called N and N. In case of mitosis, there was father's copy also, mother's copy also. Mother's copy also, father's copy also. That was in case of mitosis. Here is totally different. Now, when meiosis second will occur, meiosis second I told it is same. Now there will be meiosis second. Meiosis second is like same mitosis. Same will happen. What will happen? That now the arrangement will be same. See, now this part will be in the one. Firstly there will be same prophase, not like this stage. In case of mitosis, you have seen there is prophase, metaphase, anaphase, telophase. Now you can see the mitosis lectures. Same will occur in mitosis. What occurs in the mitosis, same will occur in case of meiosis first. And there will be the formation of four cells.